For this video, we're going to be talking about AP Biology or College Biology, Unit 3, Cellular Energetics, but we're going to be focusing in on Section 3.3, which are factors that affect enzyme action. Please just note as a disclaimer below what we have stated, and let's get started with understanding a little bit more about those factors. So there's a big word that you need to understand when it comes to Section 3.1 all the way up to 3.3, which is the term denaturing. And you might be thinking, okay, what exactly is denaturing and what does it have to do with enzymes? The thing is, we're going to focus in on Unit 1 a little bit and see if you remember some things that you learned there. So in Unit 1, if you remember, you learned about different macromolecules. And one of the things you've learned about is protein. Just so you're aware, enzymes are a type of protein. And if you remember, the thing is, proteins have bonds that allow it to form not primary, not secondary, but the tertiary structure. So 3D structure or that tertiary structure has been folded. So if it's folded, it's favorable. That's what we like. That's what we want. And what happens is when we denature, that means that we add some heat as a source or some sort of denaturing mechanism to take that folded protein and allow it to break down. Therefore, it becomes unfolded. Now, when it comes to proteins, specifically enzymes, if you remember, we talked about the way enzymes work is you have to be able to take the enzyme, which we're going to represent with the letter E, and you have to have the substrate attached together at the active site. And the way that they bind together allows for the enzyme to then take the reactants and turn it into the products that we need. It basically affects the reaction rate at which the reactant turns into the product. But that only works if the substrate can attach to the enzyme properly. See, the thing is, when we have denaturing, what ends up happening is the substrate is not going to be able to attach to the enzyme properly at that active site because the active site or the region in which it attaches to is now damaged. So it can't bind properly, which means that you could have enzymes there, but they're not actually working effectively. So that's what denaturing does. Let's talk about environmental factors that influence the way that these reactions do or do not work. Now, I put a picture of a graph here because if you are given a free response question or a multiple choice question, oftentimes you're expected to look at a graph or a table and decipher what it means when it comes to enzymes. See, the thing is, when you have enzymes, they can possess what's called an optimum temperature, which means this is the temperature at which the enzyme works the best. So, for example, if you take a look at the picture over here and the graph, you can see that the reaction rate tends to go up, and that's when the temperature tends to increase. However, when it reaches around like 47 degrees here, you could see that it tends to plummet. The reaction rate plummets. So what happens is the optimal temperature for this specific enzyme works best at 47 degrees. So we could say that at that point, at around 47 degrees, is when it's going to denature, and that enzyme is not going to work effectively. So that's how temperature affects an enzyme. Now, there could be various different types of enzymes that work at various different temperatures that are optimal for itself. So just be aware of that factor. Now, let's take a look at another factor on enzymes, which is pH. So if we take a look at the graph here, I just wanted to show that you still have that same bell curve shape that we saw in the temperature, where each enzyme, there is one enzyme here, another enzyme here, and another enzyme here, and they peak at different pHs. So if you look down here, this one is around 3, this one is around 7, and this one is around 10. During these points, are when the enzymes are going to work the best. And then afterwards, it sort of goes down. Now, just like the other graph, I wanted to show you this graph because they tend to give graph-related prompts, specifically for FRQ, and you're expected to decipher it. And I just wanted to show you that both temperature and pH can peak in this way. And if you see these conditions, you immediately should know that they're talking about some sort of optimal condition in which the enzyme exists before plummeting. There's some sort of environmental enforce, or I should say force in general, to just influence the enzyme behavior. Now, let's take a look at enzyme concentration in general. Now, I want you to take a look at this graph, which seems to be very complicated, but we are going to take it one step at a time. The reason why I wanted to create this combined effect graph is because I wanted you to see how the different behaviors exist for enzymes. So, for example, if you look at the orange over here, this is just a straight line that is a line and it goes straight up. And if you notice there, what's happening is we're only taking a look at what a reaction does if there is no enzyme. So if there is no enzyme, this is what's going to occur where it just goes straight up. Now, if you take a look at the different situations in which concentration can affect, let's take a look at if we add a competitive versus non-competitive inhibitor. Just to recap, remember, the enzyme has to attach to the substrate, right? But if we have a competitive inhibitor, we're saying that, okay, we have this enzyme and it can attach, right, to the substrate. So maybe you have the substrate here, but a competitive inhibitor is also competing for that same spot. So it really is dependent on that concentration of enzymes to ensure that the substrate does or is able to bind properly. And the perfect line for that is this purple dotted line here, where it will start working and sort of taper off. And that's because of the behavior of the competitive inhibitor, which will block the substrate from attaching. Now, with a non-competitive inhibitor, remember, a non-competitive inhibitor would be the fact that you have a substrate and you have a non-competitive inhibitor that will attach to a spot on the enzyme outside of the active site, rendering this whole enzyme useless. And that works far more strategically in the sense that you have this like green line here where it's going to peak very quickly, starts working, and then it sort of tapers off a lot faster. Because as much as you can have the substrate attach to the enzyme, if the non-competitive inhibitor gets there first, it renders the enzyme useless. So that's the behavior of both of those. Now, if you take a look at the behavior of without inhibitor at all, and you just have an enzyme, it's going to work to a certain extent and then tapers off as well, but works best in the scenario of all of the inhibitors or without inhibitor. So I just want to point that out in case they give you a graph, usually in FRQs, to determine the relationships between the enzyme and concentration. Basically, increasing the enzyme concentration is beneficial because it gives more opportunity for that substrate to attach. So the more enzymes that we have, the better, regardless of whether or not there is a competitive or non-competitive inhibitor, because it gives the substrate the opportunity to bind more efficiently. However, if substrates become scarce, what ends up happening is no matter how much enzymes we increase, if we don't have a substrate that can attach, really there is no way that the enzyme will increase the activation energy. It just will be no effect whatsoever. But we also note that if there is a substrate concentration that goes beyond the point, so if we have a lot of substrates and we have a little bit of enzyme, it doesn't matter how many substrates we add. If we don't have enough enzymes, then we can't attach, and therefore it doesn't matter how many enzymes we have, it's still not going to work because it's dependent on the fact that you have enough enzymes for enough substrates. So having enough enzymes and having enough substrates works effectively if you don't have any of those confounding variables, meaning you don't have the competitive and non-competitive inhibitor that affects the behavior. So I just wanted to point that out for reaction rates. Now let's try to do a quiz question together. The thing is, unlike previous questions, I wanted to focus on an open-ended question so you can see or highlight the way you want to think outside the box for potential free responsive questions. Take a moment, pause this video and unpause once completed, and let's discuss together. Okay, now that you're done reading the question, let's review the question together. It states, in light of the fact that enzymes thrive under particular optimal conditions such as temperature and pH, how could shifting tides of environmental changes such as global warming or the acidification of ecosystems such as the oceans and soils influence the delicate dance of enzyme activity within living organisms and their habitats? While this may not be a free response question, this allows you to think outside the box. So let's try to review possible scenarios in which you can respond to this question. The thing is, you want to bring together firstly global warming. Remember, we talked about in the previous pictures the graph in which we have the optimal conditions. There is a certain point in which enzymes will work at higher temperatures before there is some sort of denaturing, which therefore can decrease. So global warming, if there's enzymes in things like plants, we will have issues with enzymes that work properly in plants, which need certain types of enzymes that work in certain temperatures, and same thing with animals like humans. We are going to focus on photosynthesis in later chapters, specifically this unit, but later on in a different section. So just note that photosynthesis is a process that's associated with plants. Now, if we talk about pollution and acidification, you want to think of pollution based on the question to state that it increases pH in some way, or it could decrease pH in some way. Remember, acid is below the 7 number. Remember, pH goes from 1 to 14, and if you're less than 7, you are more acidic. So you can tend to say that if you're going lower in pH, this can also impact the way the enzyme works because different types of enzymes have different pH levels that work in optimal conditions. Now, there could be adaptation that exists. Like you can think of the way we talked about this idea that you have different inhibitors, and there could be an inhibitor that could be placed in some sort of medication, for example, that could shift the way that the enzyme works. And that could be a potential solution. Another thing could be changing the type of elements, such as carbon, in your environment. Please note that this is not a true free response question, but a potential subset question that you could see in a free response question and potential solutions that you could be provided. I hope you like that review. Make sure to subscribe, hit that like button. Hey, Geeking Out on STEM family, guess what? We want you to hit that subscribe button. Why? Because we'll be producing more content just like this for the AP Biology exam and the new digital SAT exam. By subscribing, you'll be getting exclusive, up-to-date tips, tricks, and secrets for both exams. Also, if you're a parent, a teacher, a homeschool instructor, or a student, you might be looking for affordable and exclusive digital products to help improve multiple choice questions, free response questions, or question bank questions related to both the digital SAT or AP Biology exam. We have them here. Make sure to check out our Linktree in our bio for curated resources that you won't find anywhere else. These unique resources are designed to help you raise your score. By hitting the subscribe button, the more subscribers we get, by the way, the more videos we can make. So make sure to join our family to ensure that you get a perfect score for both exams. We can't wait to help you succeed.